Now that so now that we have an understanding of the anatomy of plants, we're going to talk about some actual processes, so how things work, and specifically how minerals, water, and sugars are transported through the plants. So first, a little bit of review. So if you remember, when we were first talking about plants, they have adaptations for land as they evolved from ancestors that came out of the water. So they have roots and leaves, which are for absorption and photosynthesis. They have a lignified vascular system, which allows them to stand upright and grow vertically and also hold their xylem and phloem. Then they also have sporopollen, which is a polymer that prevents desiccation. So plants have this dual challenge of balancing the needs of photosynthesis with preventing water loss. Too much water loss, of course, results in cells that no longer function. And so, but water is needed to move through a plant, and the evaporation of water is dependent on that process. So we'll talk about this balance and what plants do to use and then prevent the overuse of these systems. So photosynthesis, as you remember, is a combination of carbon dioxide and water and energy from sunlight to make sugars and a byproduct of oxygen. So to get carbon dioxide and to trap energy from the sun, this requires exposure to the air. You have to, because that's where the carbon dioxide is coming from. Exposure to the air, however, also exposes the plant to water loss and dehydration. And sunlight, which has energy, also causes water to evaporate. So you also have water loss. So this then is the balance that plants have to deal with. Photosynthesis, however, is the way that plants get energy. So plants will expose their leaves, depending on the area, at different arrangements. And this is called phyllotaxy, the arrangement of the leaves on a stem. Now, if a plant wants to increase the surface area of the leaf and its exposure to the sun, it will arrange its leaves accordingly. So most angiosperms that are trying to maximize their exposure to the sun have their leaves about 137.5 degrees apart. And what that does is allows for maximum exposure. So you can look down on the leaf, you can see just down on the stem, and you can see the ground is not exposed to the sun, that all of the sun shining on the plant reaches the leaves. Now, different species have different amount of coverage, and we can measure this in what's called the leaf area index. So this plant has a greater leaf area index than this plant. So this minimizes shading, exposes more of the leaves to the light, and this shade index or leaf area index then is that measurement. Some plants in arid environments don't want to be overexposed to the sunlight, so they may have a vertical arrangement. So if you're in an arid, very hot environment, that might be that is a common strategy. In low light conditions, however, a horizontal arrangement like these leaves are ideal. So these are probably in the canopy of a rainforest where there are trees overhead, and so not a lot of sunlight gets in and gets to the ground floor, and so they are going to maximize their horizontal arrangement of leaves to capture that sunlight. There's also a trade-off between growing vertically to achieve exposure to sunlight and outcompete other plants, and growing horizontally or branching out. So any energy towards one can't be spent towards the other. And so large trees that are both vertical and branching out take a long time to grow because a lot of energy is required to do both. All right, so the roots are very vital to water and mineral transport because this is where the origination of that capture begins. But the roots also are helped by a symbiotic mutualistic relationship through mycorrhizal fungi, which help extend the roots and their their infiltration of the soil to absorb more water and more minerals. And roots also to help stabilize the plant, so they are structural in anchoring the plant in the soil. Now, the way that water and minerals move through cells from one to the other can be through different areas. So the apoplast is everything external to the plasma membrane within the cell wall. The symplast then is everything within the cell, within the plasma membrane, including plasmodesmata, which are structures that connect the membranes of two adjacent cells. And so when those are the physical structures, but these also relate to the pathways in which water and minerals will pass through. So we have the apoplastic route through the cell walls, external to the plasma membrane, and through the extracellular pathways, so you may even be outside the cell wall, and the symplastic route, which is through the cytosol. And then you also have kind of the combination of the two, which is called the transmembrane route, where it goes through the cytosol, maybe even through the storage vacuole, and then also through the cell wall, and basically just goes through both apoplastic and symplastic. All right, so the overall transfer of fluids is controlled by the permeability of the plasma membrane. So we'll have to go back to Bio 1 to remember some of the ways that water and minerals are transported. And they can be through active transport, which requires ATP. And in plants, we use a proton pump. Okay, so it uses ATP to create a gradient and put protons on the extracellular fluid. Well, now you have a concentration gradient, and these hydrogen ions want to go back into the cell, and we can take advantage of this by having a co-transport system, where as hydrogens pass through, we can pass other molecules such as neutral solutes or ions. We also have methods of diffusion and osmosis. So water will follow solutes through a plasma membrane, and in this case we have ion channels for the diffusion of ions from greater to lower concentration. And if you remember, we have facilitated diffusion, which is through one of these proteins. Aquaporins also are proteins to help facilitate diffusion. So osmotic pressure then is the measurement of of the flow of water as controlled by solutes and physical pressure. So psi, this Greek letter or Greek symbol, is used as the notation for water potential, which is the total effect of solute concentration and pressure, physical force. So psi is the additive effect of solute pressure and physical pressure, which I'm just kind of repeating this, but we can denote those by a sub S and a sub P. It's measured in megapascals, which is a unit measurement of pressure, and it determines the direction of water movement. Water flows from a high water potential to a low water potential. Turgor pressure then is an example of the force exerted from cell to cell onto protoplast, the living part of the cell, by solute concentration and physical pressure. Okay, so not looking at cells, but just setting up a semi-permeable membrane with two concentrations of solutes. You can determine, we can look at the solute and physical pressure. So here we have a hypertonic solution, and water is flowing to the other side where the solutes are. This is an example of our solute pressure. Here we have a sealed tube which actually pushes water through the semi-permeable membrane. There are no solutes involved, so this is an example of our physical pressure. In this example, we have solutes, so water is going to want to go this way, and we also are pushing with physical pressure. So here we have the physical and solute pressure equaling out. You can also have a negative pressure. So here we have a seal and are pulling up. This pulls water through the semi-permeable membrane, so we have physical pressure here as well. Now these pressures are at force in cell walls. A turgid plant cell, which is one that has is normally functioning, the physical and solute pressure are equal. So the force exerted by the cell wall equals the force exerted by the water within the cell. In a flaccid cell, so one which is wilting, the solute pressure is reduced and the physical pressure of the wall is zero. So your solute pressure is more negative than your physical pressure. And then in a plasmolyzed cell, so this is one that is no longer functioning, the solute pressure is too great, so it's much more negative than the physical pressure. And so you would have then the cell membrane pulling away from the cell wall as you are losing more and more water. Okay, so passive transport, as you remember, is a function of diffusion, osmosis, facilitated diffusion, and bulk flow, or also known as filtration. And this occurs, bulk flow, through the forcing of water into and out of cells due to the pressures which we just talked about, through xylem, which is water and solutes, through the cell types, tracheids, and vessel elements. In the phloem, you have nutrients flowing through different types of cells, our sieve tube elements, right here. And the xylem and phloem have little tissue to resist movement via pressure or bulk flow. So whatever pressures are exerted, generally from one end to the other, can move water and sugars and other things through. All right, so first we're going to talk about how water and minerals flow, which starts at the roots. So first, water and minerals are absorbed at the tips via root hairs, and then transported through the cells, either through the apoplastic or symplastic root. Okay, and this is transport from the root hair through the cortex, or the outer part. The water and minerals then have to pass through the endodermis, which has a Casparian strip through it, which blocks any apoplastic route, and so everything has to go through the Casparian strip and through the cells through the symplastic route. It then enters into the xylem through the symplastic route, and then is regulated by passive and active transport through the endodermis, and is now ready for transport through the plant, through the vessel elements and tracheids. All right, so transpiration is the use of, or sometimes called evapotranspiration, the use of evaporation to move water through the plant. And this is, like our example before, an example of negative pressure. So what happens is, at the surface of the plant, you have evaporation occurring. Okay, and evaporation pulls water out of the plant. That creates then this negative pressure on the xylem in the leaves. And that negative pressure then pulls water through the xylem, which is one continuous tube, all the way down through the roots. And so it pulls water out of the roots and then through the whole plant. And this is called bulk flow, right? So this is similar to how a straw works. You exert a negative pressure by sucking water, and then that water at the bottom is replaced and continually flows, as long as there's a negative pressure there. In transpiration, however, the sunlight is doing the sucking instead of Obama, and evaporation is creating the negative pressure. So part of this bulk flow, it's aided by what's called the cohesion tension hypothesis. So transpiration is the source, but it is aided by the cohesive properties of water. So water sticking to itself allows a continuous flow or connection between water molecules, which allows it to continue throughout the plant. Water is also adhesive, so it will stick to other things as well, including the cellulose in the xylem walls. So that counteracts the force of gravity pulling it down. And so these cohesion intensive properties reduce the effects of gravity and make a continuous connection between the water molecules, so they're pulled all the way through the xylem. Okay, so here's we'll have a couple of these diagrams. But the water first is exposed to the air and the sunlight and the heat, which causes evaporation through the stoma. Multiple stoma are called stomata. As that water is pulled out, that creates a negative pressure, so water then replaces it through the xylem into the different parts of the leaf. Okay, so altogether you have at the roots water and minerals being actively transported into the xylem through apoplastic and symplastic, and then through the Casparian strip through the symplastic root. Once it's in the xylem, you have evaporation occurring through the stomata. That evaporation creates a negative pressure, which pulls water all the way through the xylem in the plants. The cohesive and adhesive properties of water help aid and counteract the effects of gravity pulling the water down. At night, there is no or less evaporation occurring, and so sometimes, however, there's still active transport of water and minerals into the cell, exerting a positive pressure, and this will sometimes in the morning leave these droplets of xylem sap at the stomata, and this is called guttation. So leaves have a high surface area to volume ratio. They're very thin and broad. Okay, and this allows them to be exposed to light for sunlight, and like we said before, also exposes them to water loss. So stomata can open and close to prevent this water loss. Guard cells are the cells around the stomata. The stomata are the actual holes, and these guard cells are actually the mechanism by which they open and close. 95% of water loss occurs through the stomata, so some of it might come out the stems and roots. But the guard cells prevent water loss by closing. Stomata are also a function of turgor pressure, or using our properties of osmosis and hyper and hypotonic solutions. Turgid cells create an open stoma, and flaccid cells close the stoma. And this is controlled by the release of potassium ions. So if you release a lot of potassium ions, the water will follow and it will create this flaccid cell and close it. If you want to open it, you will increase the amount of potassium ions and water will follow it and it will become turgid. So hypertonic guard cells are open, hypotonic guard cells are closed. Now, stomata can be open or closed on the day or the night, depending on whether transpiration or the minimization of water loss is more important. This is triggered by light, temperature, carbon dioxide, and just the biological clock of plants. But they can also open or close in response to drought, high temperature, and wind, all of which will affect the evaporation. The mechanism is through a hormone called abscisic acid. Too much water is lost and not replaced through the stomata. The water lost through the stomata, the plant will wilt because the cells are flaccid and eventually will plasmalize. Transpiration can also provide temperature control, so it can prevent, it will take heat with it as that water is evaporated, so it can cool down the plant, similar to sweating. To prevent water loss, in areas where there are, where it's very hot and dry, the plants that grow there are called xerophytes, and some of them complete the life cycle only when it rains. So generally, when it's not raining, they will look mostly dead, but they'll still be alive. And when it rains, they will sprout leaves, they will sprout flowers, and produce seeds and do everything in a short period of time. Some of these leaf modifications can also reduce water loss, so they can have, vertical leaves, they can have thin leaves, or small leaves that are close to the plant. They can also open or close their stomata. So sometimes they will open their stomata at night and do the gas exchange then, and then close the stomata in the daytime to prevent water loss. All right, so that's water transport. Now we're going to talk about sugar transport, which occurs through the phloem. It starts at the leaves, and then, during photosynthetic periods, sugars are formed at the leaves through photosynthesis, and then are transported throughout the plant. And it occurs through the phloem in a process called translocation. If you remember, sieve tube elements were the cells in the phloem that transport sugars. The phloem sap is high in sugar, and it travels from source to sink, and this can change depending on the time of year. The source is the net producers of sugar, so in the summertime that's going to be the leaves, and the sink is going to be where you're going to store the sugar or use it. And this may be in the root, such as in carrots, or in the stem, such as in onions. It depends on the different type of plant. But in the winter time, a tree may then switch the source and sink from the roots to different areas of the leaf that may need to use it. Leaves won't generally be there in the winter, but different parts of the stem then. All right, so sugars first are made in the leaf and then travel via the symplastic or apoplastic routes into the sieve tube elements. And then the companion cells help move sugars from the symplast and the apoplast into the phloem. The first step is called Phloem Loading, and this requires the proton pump and then co-transport of sucrose. And then by this method, you can get a lot of sucrose into the phloem. Once you have a high concentration of this sucrose, you're in a process called phloem loading. Gravity and and this concentration of sugars creates a positive pressure by which everything just then naturally diffuses through the phloem and then into the sink. The water which will follow the sugar is then recycled by the xylem. Okay, so that's the full story. Water comes as it's pumped with minerals in the roots, and then evaporation fuels the negative pressure which pulls the water up out of the xylem through the plant, and it is aided by the cohesive and adhesive properties of water. Sugar is made at the leaves. Those sugars are loaded by active transport into the phloem. Then it naturally diffuses from source to sink. And that's how substances are transported through plants. The end.